In previous videos, we've learned about glycolysis, and we know glycolysis is when we take this glucose molecule and we do a bunch of chemical reactions, eventually converting that glucose molecule into pyruvate molecules. However, something important to realize is once we've done this first step of glycolysis, converting glucose into glucose 6-phosphate, this glucose 6-phosphate has a decision. It can either continue with glycolysis or it can enter new pathways, other metabolic pathways. And in this video, I want to specifically talk about the pentose phosphate pathway. So this particular intermediate of glycolysis can continue with glycolysis, but it could also potentially enter the pentose phosphate pathway. So what is this pentose phosphate pathway? Well, the first step is we take this glucose 6-phosphate, we do one quick reaction, forming this compound. But notice when we do this step catalyzed by this enzyme, we take one of these oxidized NADPs and reduce them to NADPH. So in this first step of the pentose phosphate pathway, we've already produced one of these reduced cofactors, this NADPH. Then we do another quick step, forming this compound catalyzed by another enzyme. But then once we form this compound, we do another step. And when we do this other step catalyzed by this enzyme, we form another reduced cofactor. We take one of these other NADPs and reduce them to another NADPH. So already in these first three steps of the pentose phosphate pathway, we've produced two of these reduced cofactors, two of these NADPHs. And we've also produced this 5-carbon intermediate. So already in these first few steps of the pentose phosphate pathway, we've produced a lot of really important byproducts and intermediates. And what's important is these reduced cofactors, these NADPHs, can be used for lots of different purposes. But one purpose is using these NADPHs as a reducing equivalent to donate electrons towards anabolism. So when we're doing anabolism, we're building larger macromolecules. But to build larger macromolecules, we need a source of electrons to donate to create those bonds. If we want to go through anabolism and biosynthesize larger macromolecules, we need a source of electrons to biosynthesize those macromolecules. And one of those sources of electrons are these NADPHs. They have electrons that they can donate. For example, for fatty acid synthesis, it's these NADPHs that donate electrons to biosynthesize fatty acids. But this NADPH can also be used for other purposes. For example, if we have this oxidized glutathione, we can take these electrons from NADPH and reduce and donate them to reduce this glutathione. So when this NADPH donates electrons to this glutathione to form the reduced glutathione, now this reduced glutathione can donate those electrons and act as an antioxidant. So this is pretty interesting. This is a way we can take glucose molecules, do this first step of glycolysis, now go through the pentose phosphate pathway to create these reduced NADPHs, which can donate electrons to glutathione to create this reduced glutathione to act as an antioxidant. So this is a way we can essentially biosynthesize these antioxidants to donate electrons and act as an antioxidant. And it's important to realize these NADPHs, it's not like this particular NADPH is going for anabolism and this particular NADPH is donating to these glutathiones. I'm just saying in general, these are two common features of NADPHs, functions that these NADPHs can do. But we also produce this 5-carbon intermediate. So this 5-carbon intermediate, we can do some modifications to essentially create the ribose sugars that we need for nucleic acids. We know nucleic acids are made out of nucleotides, and those nucleotides have a ribose sugar. They have those phosphate functional groups, and then we have that nitrogenous base. But in order to create these nucleic acids, we need this ribose sugar, and we get the carbons for this ribose sugar from this particular intermediate. So that's another reason why the pentose phosphate pathway is important. It's important for nucleic acid synthesis, to biosynthesize these nucleic acids that we know are really important for DNA and RNA and other purposes. So again, we've already seen in these first few steps of the pentose phosphate pathway, we've created a lot of important byproducts and intermediates. But we can go a step further. Essentially, remember this 5-carbon intermediate? We're creating more. We're doing a lot of these pentose phosphate pathways, so we're creating a lot of these 5-carbon intermediates. And instead of drawing the entire structure, let's just refer to them as these 5-carbon intermediates. We can take them and we can react them with each other. We can take two of these 5-carbon intermediates and react them with each other. And when we do that, we form a 3-carbon intermediate and a 7-carbon intermediate. And that makes sense because originally we had 10 carbons and we still have 10 carbons, conservation of mass. So again, we go through this step creating a 3-carbon intermediate and a 7-carbon intermediate. Now we do another reaction, reacting them again, forming now a 4-carbon intermediate and a 6-carbon intermediate. And now once we have this 4-carbon intermediate, we take another one of these 5-carbon intermediates that we produce, do some quick modifications, and now we have another one of those 5-carbon intermediates. So we react one of these 5-carbon intermediates with this particular 4-carbon intermediate through another reaction, forming again a 3-carbon and a 6-carbon intermediate. But the point is, as we do the pentose phosphate pathway, we're creating a lot of these reduced NADPHs, which we know are really important for anabolism and for antioxidant function. And again, we know these 5-carbon intermediates can be used for nucleic acid synthesis, but we know we can react them through all these complex chemical reactions. But we don't want these complex chemical reactions to go to waste, so essentially what we can do is we can salvage these carbons. We can take these carbon intermediates and essentially do modifications to them to create intermediates that can enter glycolysis. So now these carbons aren't going to waste. Now they can enter glycolysis and be used in glycolysis to create ATP and again to create these intermediates. So this is a way we can constantly do the pentose phosphate pathway to create these reduced cofactors for anabolism and for antioxidant synthesis, biosynthesizing these antioxidants, so we can keep on doing this process without wasting all these carbon intermediates. We can, again, funnel them back into glycolysis. But again, this is the pentose phosphate pathway, and it's important to realize there's 2 phases. There's this oxidative phase where we do redox reactions, where again, we're reducing compounds, so we have this oxidative phase. And then we have this non-oxidative phase. And again, the way I like to think about it is as we do the pentose phosphate pathway, we do the oxidative phase to create these really important reduced cofactors, but then we're left with these carbon intermediates, which we don't want to go to waste, so we do some modifications to them, so now we can create these carbon intermediates, which we can do more modifications to enter glycolysis, and now they don't go to waste, and they can just continue with glycolysis.